Hello guys, today's project will be the cell cycle and it's by Ramirez, and so let's begin. Okay, so the cell cycle. The cell cycle is a process that a cell undergoes to replicate or to make a new cell. And so when the cell goes through this phase, to this program, whatever, it has phases. So in G1, the cell grows. It grows and becomes bigger, and that's most of it. In the S phase, which stands for synthesis, the DNA begins to replicate. So as you see, each little edge right here, there's little edges right here and here and here, and each one, it's beginning to make a copy of itself, or take off one side of the DNA, and then later on it will replace it with another, and at the end it will come out with two strands of DNA. Then goes G2, which also does more growing. Later on comes the M phase. In the M phase, is basically, it stands for mitosis. And in mitosis, this is where the cell actually begins to separate. And later on, when mitosis finishes, then there will be two different cells. So G1 through G2, which includes G1, S phase, and G2, those are the that's called interphase. And now mitosis, I'll explain mitosis for you guys. This part. Okay. So basically, right before mitosis begins, this is what the cell will look like. And that's basically pre-mitosis, I'll call it. And that's what the cells is, a little more condensed, and it's beginning to be able, the chromosomes are beginning to be able to be seen. Now we begin prophase. In prophase, the chromosomes condense and become more visible, as you see. The DNA wraps tightly around the histone proteins, and the nucleus dissolves. Basically, as you see, now you can actually see the chromosomes. Now later on, on the next stage, and this stage is called a metaphase. In metaphase, the chromosomes line up in the middle, and organelles begin to build up and are starting to build up, like stuff like that. So basically, these are the centrioles. This right here, that right there is called the centriole. Here, I'll mark it for you guys. That's the centriole. These lines that the centriole attaches to the chromosomes, those are called spindle fibers. And then these are just the normal chromosomes. Next one. This is anaphase. In anaphase, sister chromatids, these and these correspond with these. So these are called sister chromatids. Since they together make the chromosome. Now when they are separated, they are the sister chromatids because they correspond with each other. As you've seen, by corresponding, I mean they are the same color. So in my case, they are the same color. So these sister chromatids are all pulled apart and they move towards the opposite side. And this is all done when the centriole is pulling the sister chromatids apart with the spindle fibers. Now, so, give 5 seconds, 5. So, this stage is called telophase. In this stage, the cell starts to constrict in the middle, meaning it starts to pinch in, as you see right there, and a new nuclei is formed. As you see, as I have this dotted line, spotted line, that is the nucleus beginning to form around the chromosomes. So this stage is called telophase. And then the whole cell cycle ends with the stage cytokinesis. In cytokinesis, this is what cytokinesis looks like. And cytokinesis basically is when the cell actually splits. It's actual cell splitting. So now there are two separate cells with a full set of DNA. DNA. So now they all have chromosomes and will go on later to replicate again. And again, these will later on the cells will then later become more uncondensed and become darker again and end up looking like this again and go through a whole new other cell cycle. Okay, good. So, the question is, what can go wrong in the cell cycle? Well, actually, many things can go wrong during the cell cycle. They normally function to accelerate cell division and growth during the cell cycle, but when they are mutated, they can act like gas pedals stuck with a 100 pound thing on the pedal, and basically it won't stop. This can be demonstrated by this image. In this image, the cell has grown exaggeratedly and it is too big. When it is too big, it can go on and cause cancer. So this is one way that the cell cycle can go wrong. Another way that the cell can go wrong is through tumor suppressor genes. An example of a tumor suppressor gene is p53, which triggers apoptosis, which is a self-destruct program. So right here, this is a normal cell, but say that this one is infected. This thing is infected, and it's like, oh no. And then it spreads on to this one, and later on spreads to this one. So basically, the whole cell is like bad. So what does the cell do? It's gonna initiate p53. Dang it, whatever. It's gonna initiate p53. Boom! It's gonna explode. It's gonna pop. It's gonna self-destruct. But if this can happen, wow, this one fell too. Okay. If this can't happen, this thing is gonna continue to infect other cells, and that later is gonna create a tumor. And tumor is basically bad and can also lead to cancer. So that is how tumor suppressor genes can go wrong and affect the cell cycle. Now another example of how something can go the cell cycle can go wrong is DNA repair genes. And normally they function to fix minor damage in the DNA when it's replicated, but when mutated, the DNA damages accumulate, and basically it can lead to cancer. So an example of that is saying, an analogy basically of that is like a stupid mechanic. So as you see, if this thing is broken right here, you're gonna want the right cells. But when these are mutated, these tools are not working right. They're all bad. They're acting really bad, and they can't even like work well. So then, what's gonna happen? This is broken. So then, and it's not being fixed. So then, this one's gonna break, and this is gonna break, and then everything's gonna break again. But yet again, it's gonna all break. And not break, but like it's all gonna be affected, and this is an example of how it then leads to cancer. Okay, so there might have been some parts that you might be a little confused about, and I forgot to explain that what were. So as you see in this, in every cell there is a phospholipid bilayer on the top, and that's basically the nuclear envelope, which is also right here surrounding this part. So right here. So it's right here. It's a phospholipid bilayer. It's the thing that surrounds the striped part. So it's right here. I'll highlight it. This is that circle that I just highlighted. That's the nuclear envelope. Sorry. And another part, as sorry, so another thing that you might have been that I might have forgotten to mention was the centromere. The centromere, this is a chromosome with two sister chromatids, and the two sister chromatids are connected by a centromere. The centromere is this black dot right here that I am pointing at, and that is a centromere. Sorry if I didn't explain it before. Okay, guys. So, in another way, if I forgot to mention something again, the cell cycle has checkpoints. So the cell cycle checkpoints are basically, so there might be a checkpoint here, a checkpoint here, a checkpoint here. These checkpoints are to make sure that the cell cycle is moving smoothly. So when G1 phase is finished, there'll be a checkpoint to make sure that the cell did everything correctly when growing. During S phase, there might be a checkpoint to make sure that the DNA did replicate correctly, and so that there is no mutated DNA or damaged DNA that will later on lead to a mutated cell, which could later on lead on to cancer. So we don't want cancer. We want good cells replicating. Replicate. So, and then G2 would be a checkpoint to make sure that everything through interface went correctly, and it is so that it is ready to go through mitosis and that everything runs smoothly again. So that's what cell cycle checkpoints do. And then the regulators are the ones that actually do the hard work and get the job done. As you can also see, there is another phase called G0. And G0 also acts kind of like a tumor suppressor gene, because G0 is like a break. It's like a timeout. It's the time that if the cell didn't quite grow correctly, or is already having problems during G1 or anywhere else during the cell cycle, in any other phase, it can go to G0, which is a moment where it's like a pause, where there's no growing or replicating happen. So it gives it a chance for like DNA repair genes to come and fix all the minor damages, or any other repair genes to fix the problems. And if it does not end up fixing, it will undergo apoptosis. So that is what the G0 phase is. It's just a time out to get things straight and so I can later on and continue the phase, the cell cycle. So usually when mutated cells are not stopped by apoptosis or tumor suppressor genes or alkaline genes or repair genes, that leads to cancer. And cancer is a collection, or not a tumor is a collection of mutated mutated cells. And cancer is basically really bad for the body, and it can cause you death. And it can cause death. Yes, it can. So yeah, cancer is deadly. And things like mutated oncogenes or mutated tumor suppressor genes or mutated DNA repair genes, all these things that can help prevent cancer, when they are mutated, they make it so that cancer can keep on going, and that's why it is bad. And that's basically what cancer does to the cells and its defenses during the cell reproduction phase. So that is cancer. Usually when we have 23 chromosomes from each of our parents, so we have 46 in total. So basically that is what the diploid cells are. That means that we have 23 from mom and 23 from dad. So that's why there's 2, because we have 2 parents and we receive 2 sets of chromosomes from each parent. So that's what a diploid cell is. Diploid cells.